Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss G-protein coupled receptors. In the previous video, we talked about the ligand-gated ion channel receptors, and we discussed three examples: the NACHR that binds acetylcholine as a signal, the NMDA-AMPA receptor, which binds glutamate, and the glycine receptor that binds glycine. So today we'll focus on GPCRs. This is the most common type of receptors that we find in the body. And we'll talk about the GPCRs that are associated with the G-alpha S protein. Here we'll use the glucagon receptor as an example, which binds glucagon. And then we'll look at the GPCR that's associated with G-alpha I. Here we'll look at the M2, M4 acetylcholine receptors that bind acetylcholine. And then finally we'll look at the GPCR that's associated with the G-alpha Q protein. And here the example we'll use is the M1, M3 acetylcholine receptors that again binds acetylcholine. First, let's look at the GPCR that's associated with the G-alpha S protein. And we discussed this in a previous video, the glucagon receptor that binds the signal glucagon. As discussed in a previous video, during starvation, the hormone glucagon that comes from the pancreatic alpha cells will increase in concentration, and they will bind glucagon receptors on the target cells. Now, these glucagon receptors are G-protein coupled receptors. So you can see this is the glucagon receptor, and associated with this protein is a G protein. Now the G protein is, you can see, stands for guanine nucleotide binding protein. That's where the G comes from. These proteins belong to the GTPase family of proteins. It is a trimer. It has three subunits: the alpha subunit, the beta subunit, and the gamma subunit. So we see the alpha subunit, the beta subunit, and the gamma subunit. The alpha subunit here, this subunit, can be of three types. So we could have G-alpha S. This will stimulate the cyclic AMP dependent pathway. Then we have G-alpha I. If the receptor is associated with G-alpha I, then that would inhibit the cyclic AMP dependent pathway. And if it's associated with the G-alpha Q protein, then it will stimulate another pathway, which we'll talk about in a few minutes, and that's the inositol phosphate pathway. So, depending on the G-alpha protein that is associated with the receptor, we could have different pathways that could be stimulated or inhibited. Now, the glucagon receptor is associated with the G-alpha S G protein. So normally the G-alpha S has a GDP nucleotide, a GTP nucleotide that is bound. And when the GTP nucleotide is bound, this protein is inactive. However, when the signal binds the receptor changes shape, and that causes the G protein to change shape. The G-alpha S changes conformation. The alpha S subunit will now dissociate from the G trimer, and now the GDP will be replaced by GTP. So now we have GTP bound to the G-alpha S, and this is going to activate the G-alpha S. Now the G-alpha S also has an intrinsic GTPase activity. So gradually over time, this GTP will slowly be hydrolyzed back to GDP. And as the GTP is replaced by GTP, the G-alpha S protein slowly becomes inactive again after the signaling is done. So this gradual hydrolysis of the GTP back into GDP, making the G-alpha S protein from the active form back into the inactive form, ensures that the signaling doesn't keep on happening. It gradually is terminated. Okay, so now the G-alpha S protein has a GTP bound to it, is activated. So what does it do? It will go and bind to the enzyme adenyl cyclase, and it will activate this enzyme adenyl cyclase, and this enzyme will now convert ATP into cyclic AMP. So ATP substrate will be converted into cyclic AMP by this enzyme adenyl cyclase. So this means the concentration of cyclic AMP gradually increases in the cell. Now cyclic AMP is a second messenger, it's sort of an intracellular hormone. Of course, the first messenger is the signal, in this case glucagon. As we discussed in the previous video, cyclic AMP will now go and allosterically activate another protein called protein kinase A. And protein kinase A is located in different parts of the cell depending on its attachment with the protein called A anchoring protein, or ACAP. So here in this figure, we see a pKa being held together by ACAP at the plasma membrane. Okay, so the ACAP can have this pKa located in different parts of the cell. So in this case, the pKa is held at the plasma membrane by the protein AKAP or ACAP. Anyway, as we discussed in the previous video on starvation, what the activated pKa will then go on to affect many other proteins, which will eventually result in several of these effects that we saw earlier: increased gluconeogenesis, decreased glycolysis, increased glycogen breakdown, decreased glycogen synthesis, increased fat breakdown, as well as increased amino acid catabolism. So one of the most important biological outcomes of this, if this cell was the liver, would be the liver cells would now start exporting glucose into the bloodstream by increasing glycogen breakdown and increasing glucose synthesis by gluconeogenesis. And this is very important in starvation because this is how we maintain our blood glucose concentration when we are not eating during starvation. There are several other GPCRs that are associated with G-alpha S proteins. Here are a few examples. Prostaglandin PGE2 is the signal that binds the receptor called E-prostanoid receptor that's involved in the renin-angiotensin system. This is something we'll talk about in a future video when we look at the biochemistry of various physiological processes. Then we have thyroid-stimulating hormone, TSH, is a signal that binds the TSH receptor. Then we have the hormone ACTH, which is a signal that binds melanocortin 2 receptors or MC2R receptors. This results in a release of cortisol by the renal glands. So all of these are all examples of GPCRs that are associated with the G-alpha S protein, and they all will work in a very similar mechanism as what we described earlier for glucagon receptor. So this we'll talk about in future videos when we come across this various physiological processes such as the very important renin-angiotensin system, or when we talk about the thyroid gland, or when we talk about the adrenal gland. Now let's look at an example of a GPCR that is not associated with G-alpha-S, but now with the G-alpha-I protein. And examples of these receptors would be the acetylcholine receptor, which is of type M2 or M4, where the signal is acetylcholine. So let's look at an example of this. So we have this M2, what we call muscarinic receptors of the M2 type. And we do find these receptors, for example, in the sinoatrial as well as the atrioventricular nodes of the heart. These regions are very important, have very important pacemaker functions. Basically, they control the heart rate. These AV nodes and SA nodes, and we'll talk about this under heart biochemistry, they control the heart rate. So these cells here have these M2 muscarinic receptor type 2 receptors on the surface of the cells. So when the signal acetylcholine binds to these M2 receptors, and these M2 receptors are associated with G-alpha-I, what happens is the G-alpha-I, shown here in this purple color, this protein now, instead of stimulating adenyl cyclase, it will inhibit adenyl cyclase. Okay, so it will do something very different from what we talked about earlier. So adenyl cyclase will be inhibited, pKa will not be activated. So we have very different biological outcomes. One of the effects of this G-alpha protein is also that this protein affects potassium channels in the AV node as well as the SA node. And by affecting these potassium channels is going to cause hyperpolarization. And that means the heart rate is going to slow down. Okay, so the end result of acetylcholine binding to the M2 receptors on these cells here, at the AV node and the SA node, is it will decrease the heart rate, what we call bradycardia. Other examples of GPCRs that are associated with G-alpha-I protein include ghrelin, which binds the ghrelin receptor. We talked about it in a previous video. Norepinephrine, that binds the alpha-2 adrenal receptor, as well as the endocannabinoids such as anandamide and 2-arachidonyl glycerol. These signals bind the cannabinoid receptors such as CB1 and CB2, and these are involved in various functions including pain, mood, appetite, as well as immunity. Now let's look at a GPCR that is associated with this G-alpha-Q protein. And here we're going to talk about the example of the cholinergic receptors, M1 and M3, that bind acetylcholine. So here we have the signal, and let's say this signal was acetylcholine, and this is a receptor. This would be your M3 receptor, and let's say this cell was a smooth muscle cell of the bronchiole. Okay, smooth muscle cell of the bronchiole. So in this case, we first see the acetylcholine will bind to the receptor, and this is the G-alpha-Q associated receptor. So we see a G-alpha-Q, and as before, the GDP in this protein will be replaced by a GTP. So the G alpha Q protein that is associated with a GTP now becomes activated, and it will bind this enzyme called phospholipase C. So once this enzyme becomes activated by the GQ protein, this enzyme now will catalyze the breakdown of the substrate here. This is called phosphatidyl inositol 4,5-bisphosphate, or PIP2. This PIP2 will be broken into two parts. It will form this part here, which is diacylglycerol, that is hydrophobic and that stays in the membrane. And the second part is this part here, which is IP3, or inositol 1,4-triphosphate. Now, since there are so many phosphates here, remember phosphates are negatively charged, this part is very water soluble, so this goes into the cytoplasm. So this part remains in the plasma membrane, the DAG. The IP3, being very water soluble, enters the cytoplasm. The IP3 will now go to the endoplasmic reticulum, and there it will open up calcium channels. So there's a lot of calcium stored in the ER. So in the ER we have a lot of calcium. So now these calcium channels will be opened by IP3. So now the calcium will travel out of the ER into the cytoplasm. So now the cytoplasm will have a high concentration of calcium. So this calcium that is in the cytoplasm at a high concentration will now go and activate this enzyme called protein kinase C, or PKC. Meanwhile, the second product that is formed from the phospholipase C catalysis, which is diacylglycerol, this will also go and activate protein kinase C. So not only does the calcium activate protein kinase C, the DAG also will activate protein kinase C. Now, once protein kinase C is active, this protein will now go and phosphorylate and activate many other proteins, including various enzymes, nuclear proteins, as well as cytoskeletal proteins. There are approximately 13 or so isoenzymes of protein kinase C, which have different tissue distribution patterns, target protein specificity, as well as biological roles. Now, calcium levels, as it increases in the cell, in the cytoplasm, the calcium will also bind a protein known as calmodulin, and this calmodulin will then undergo a change in shape. Calmodulin is associated with, or is part of, numerous other proteins and enzymes such as calcium calmodulin dependent protein kinases, or CAM kinases, adenyl cyclases in the brain, calcium release channels in sarcoplasmic reticulum, CAMP phosphodiesterase, glutamate decarboxylase, nitric oxide synthase, as well as plasma membrane calcium pumps. So the binding of calcium to calmodulin will affect all of these proteins that are associated with calmodulin, and that's going to result in all kinds of biological responses in different cell types. So if this cell was the smooth muscle cell of a bronchiole, then what would happen is the binding of acetylcholine to the M3 receptor would lead to increased calcium concentration, and that calcium will cause the muscle cell to contract. And when the smooth muscle cells of the bronchioles contract, that will lead to bronchoconstriction. There are several other examples of GPCRs that are associated with the G-alpha Q protein, and that includes angiotensin receptors, gonadotropin releasing hormone receptors, histamine receptors, oxytocin receptors, platelet-derived growth factor receptors, serotonin receptors, thyrotropin releasing hormone receptors, as well as vasopressin receptors. And again, we will talk about the biochemistry of all these processes and these receptors in future videos. So hopefully this discussion with examples give you a good idea of the structure as well as functioning of the GPCRs. In the next video, we shall look at enzyme-linked receptors that have intrinsic enzyme activity, as well as those that have associated enzyme activity. So until the next video, thank you very much for watching, and have a wonderful day, and we'll see you next time.